photosynthesis. Let's get into this discussion of sunlight and transfer of energy. Photosynthesis. Let's get into this process of chemical reactions.
Photosynthesis, a process in which plants convert sunlight into sugar to use as food, is not as simple as it may sound. It is actually a combination of two light reactions that take 6 molecules of carbon dioxide and 6 molecules of water and create 1 glucose and 6 oxygen molecules. This all takes place inside the chloroplast of the plant cells. The stacks you see are many thylakoids in a group called a granum. This is where the first process takes place, the light-dependent reaction. This process requires sunlight in order to take place. The chlorophyll and the thylakoid membrane take in the light energy and release an electron. In order to replace this electron so the process can continue in the future, water molecules break their bonds, which are electrons, to fill in the gap. This also leaves oxygen molecules and positively charged hydrogen atoms. Meanwhile, the electron travels through the membrane and ADP takes some of its energy and stores it, transforming it into ATP. The electron continues and NADP+ takes it and the hydrogen atom to form NADPH. These two are crucial in the next process, the light-independent reaction, or Calvin cycle. The light-independent reaction takes place in the stroma of the chloroplast, the area all around the granums. This process does not need light because the energy it needs is in the ATP, and the NADPH acts as an electron carrier with the extra electron connecting to the hydrogen atom. However, carbon dioxide is needed to complete this series of anabolic reactions that create larger molecules from smaller molecules. A complex series of small reactions takes these 3 reactants and turns them into formaldehyde, which later turns into glucose. It also causes the ATP to revert to ADP because it used the energy that it had stored, and causes NADPH to revert to NADP+ because it took the electron and hydrogen in the process.
Uracil is an inhibitor of photosynthesis. It is used primarily in agriculture to stop the growth of unsavory plant life, but can also be found making enzymes in the body and in pharmaceuticals that stop the reproduction of cancerous cells. Chemical engineers can be found making pesticides that can be shipped out to farms, and farms can be seen using those pesticides and herbicides. Uracil stops the process of photosynthesis by taking the D1 proteins that make up the herbicide and inserting them into the chloroplast thylakoid. These proteins block electron transportation within the thylakoid membrane, which eventually stops the fixation and production of CO2 and energy needed for photosynthesis. Plants do not die off of herbicides and pesticides, but from indirect effects on other processes. The uracil herbicide is the spark of depletion, causing other destructive reactions to take place. Cellular respiration is the process of making ATP from glucose. When an animal eats a plant, the plant's glucose needs to be converted into ATP, which is an energy form used to complete work. ATP is used as an energy source in order for a cell to complete mechanical, chemical, or transport work. Photosynthesis and cellular respiration are very similar in that they both convert one type of energy into another. Because cellular respiration is the exact opposite process of photosynthesis, you can literally switch to the balanced equations reactants and products from photosynthesis to get the equation for cellular respiration. While photosynthesis turns 6 molecules of carbon dioxide and water into 1 glucose and 6 oxygen molecules, cellular respiration takes the 1 glucose and 6 oxygen molecules and makes them into 6 carbon dioxide and 6 water molecules. In order to create ATP, there are 3 processes that take place: glycolysis, Krebs cycle, and oxidative phosphorylation. Glycolysis, which is the first phase of cellular respiration, takes place in the cytoplasm and cytosol of a cell. The main purpose of glycolysis is to convert 1 glucose molecule into 2 pyruvate molecules. During the process, energy is released as glucose is being broken up, so that extra energy is absorbed by ADP to make ATP. Extra electrons are also picked up by NAD+ and making it into NADH. In the mitochondrial matrix, the Krebs cycle completes the breakdown of glucose and takes 2 pyruvate molecules and turns them into 2 acetyl-CoA molecules. Because 2 acetyl-CoA molecules are released, the cycle is happening at the same time in 2 different places. Throughout the cycle of 1 acetyl-CoA molecule, 2 carbons are released and are paired with a CO2 to make 3 CO2. Energy is taken from acetyl-CoA and combined with ADP to make ATP. An electron is given out and added to 3 NAD+ to make 3 NADH, and another electron is released and added to FAD+ to get FADH2. The next cycle is oxidative phosphorylation, which takes place in the intermembrane space of the mitochondria and is driven by electron transportation. NADH and FADH2 from the Krebs cycle come along and drop off their electrons into the protein complexes. The electrons are passed throughout different complexes, attracting hydrogen molecules at each station. Those hydrogen molecules are shot out the other side of the complex into the hydrogen gradient, or the intermembrane space. There's a high concentration of hydrogen in the intermembrane space, but a low concentration in the mitochondrial matrix, and this makes the hydrogen molecules want to move from a high to low concentration. Hydrogens move through the ATP synthase in order to get back into the mitochondrial matrix. When moving through the ATP synthase, the hydrogens move so quickly that kinetic energy is created. That energy is absorbed by extra ADP and is turned into ATP. This is the main source of ATP as it makes the most in the shortest amount of time. The hydrogen molecules are either sent through the same process or taken in by an oxygen molecule and an electron that was retrieved from the protein complex. Water is labeled a byproduct of oxidative phosphorylation because it's not what we were trying to make in the first place, but it came along anyway.
Pyruvate kinase deficiency is a metabolic disorder that is caused by a mutation to the PKLR gene that lowers its ability to produce pyruvate kinase. This mutation can occur in the person regularly or be passed down genetically if both parents have the recessive gene. PK is needed in the final step of glycolysis to finish breaking down glucose into pyruvate. Without enough PK in the cell, ATP production decreases and the cell loses energy. It is then unable to carry out reactions within itself and it dies prematurely. Among other symptoms of PK deficiency are anemia, a decrease in the number of red blood cells, jaundice, a yellowing of the skin and whites of the eyes, and gallstones in the gallbladder. Some methods of diagnosing PK deficiency are checking for the PKLR mutation, monitoring the activity of pyruvate kinase, and assessing bilirubin levels. High levels lead to jaundice. While not all cases need to be treated, blood transfusions and splenectomies are common treatments for this disorder.